So hello everyone, good morning and welcome to my presentation. I'm going to be presenting a topic that of course all of us have heard quite a lot about in the news these last few months, coronavirus, and specifically I'd like to talk about why it's so difficult to make a vaccine for it, because as you know, this is one of the current challenges that scientists are having to face as to why it's so difficult to make a vaccine for it, because it's taking quite a long time to do. So hopefully by the end of today, you should be able to have a better idea of why that is. Okay, so let's make a start. Okay, so first of all, this is an image which you're probably familiar with. So this is what the coronavirus actually looks like, okay? Basically, any particular virus, on its surface, it's got a lot of these little key-like structures on its surface, okay? So those of you who are currently on GCSE biology or equivalent, you might have known, you might have come across the name of these key-like structures, okay? So it begins with an A, and they are called antigens, okay? So antigens are essentially an ID card that a virus has on its surface, okay? Every single virus has a particular shape of antigens. They're like little keys, okay? Like keys to open a lock, you can say. So this is a bit like the signature of a particular virus, okay? So I'm just going to quickly go through with you a little bit of a layout for what the session will cover. So number one, how your body responds to the coronavirus. Mainly, we're going to be homing in on specifically the production of antibodies. This will then lead us on to talking about how a vaccination normally works. And then we are going to embed all this together and talk about why it's therefore so difficult to make the vaccination for coronavirus. Okay, so first of all, how does the coronavirus actually affect your cells? So what actually happens is that, so here, the virus attaches itself to a receptor on one of your body cells like this, okay? And what it does is it basically inserts a piece of its genetic material, RNA, and this piece of viral RNA will go and join your own genetic material, okay? So on the chat, if anyone knows where in the cell we have our genetic material stored, where in our cells our DNA is stored, if you'd like to just write on the chat, that can be the first question of today, and I will reveal it shortly anyway. But basically, that's what happens. The virus will insert its own genetic material into our cell, and then it will join our own genetic material, which is located in, well done to those of you who guessed, in our nucleus, okay? Our nucleus contains all our DNA. Well, of course, there's a little bit of mitochondrial DNA located in the mitochondria, but the majority of our DNA is contained within our nucleus. And so what actually happens, I'm just going to now draw something on the screen as well. So what actually happens, okay, is that the coronavirus, it inserts its own genetic material into our cell, and then its genetic material will join our genetic material like this, okay? Now, as you probably will have covered in your courses, what the body does with its own genetic material is it copies it into proteins, okay? Now, if anyone knows any examples of proteins, please feel free to write them in the chat, but I can give you some as well. For example, enzymes are made of protein, some hormones are made of protein, our muscles are made of protein quite heavily, and that's why you get things like protein shakes, etc. So basically, our DNA is copied into our proteins. When the virus inserts its own genetic material into our cells, then what happens is that our cells will also make proteins from the viral DNA, because unfortunately, our cell thinks that this new piece of DNA is part of its own DNA, okay? Unfortunately, our cell can't tell the difference between its own DNA and viral DNA. And so essentially what our cell will do is it will make viral proteins from the piece of viral DNA that's now joined its own genetic material. And it's this viral protein that will actually then start to take over the body, okay, which is very dangerous, as you know. Okay, so how do we react to this? How does your body actually fight back? So you probably all have heard of something called the immune system. So the immune system is our system of soldiers, you can say, our defense system against anything that will invade our body, okay? Now, if anyone knows any particular cell that are involved in the immune system, there's a particular type of cell, a generic term for it, okay, which is involved in the immune system. Please could you write it onto the chat? I'll give you a clue. Red blood cells are involved in carrying oxygen. So another kind of cell is involved in the immune system. Okay, I'll reveal it in just a few moments, but if anyone does know, please do write it onto the chat. And you might have guessed in 3, 2, 1, it's your white blood cells, okay? Our white blood cells are the cells which form the majority of our immune system, and they release little proteins which we call antibodies. So you probably all have heard of antibodies, especially from the news at the moment, the antibody test, which has just been approved yesterday for the coronavirus. So basically antibodies are little proteins which are released by white blood cells, okay, to help us to get rid of the virus. Okay, so now we will now be exploring exactly how these white blood cells work and how they produce antibodies and how that relates to vaccinations and in turn why it's so difficult to make them. So in a normal circumstance like this, okay, our white blood cells are normally circulating around our body, and essentially they are looking for anything that should not be there, okay? Our white blood cells are going around our body and checking whether all the cells of our body have something called a self marker. Okay, this is a little bit like an ID card. Okay, all the cells of your body are coated with self markers. They tell your soldiers, they tell your white blood cells not to attack them because they belong to your body. They're self markers, they're self antigens. Okay. However, what if a foreign pathogen like a virus or a bacteria or a fungus enters your system? They won't have these ID cards that tell your body that those cells are from your body. So basically, these particular cells will be recognized as being non-self, okay? So any foreign virus or any foreign bacteria that will invade your body will not have these self markers. They will have foreign markers, which your white blood cells will not recognize, okay? This will tell them that they need to attack them because these are not self markers. These foreign invaders are not parts of your body. They don't have your body ID cards on their surface like all your other cells do, okay? And that is why as soon as a white blood cell comes across any foreign looking cell, it'll immediately generate an attack, which we call an immune response. Okay, so how exactly does this happen? So I'm going to compare this a little bit to like an army situation. So our body has something called the first line of defense. These are, you can say, the soldiers in the front line, okay? There's a particular kind of white blood cell that constitutes the first line of defense. These are the first white blood cells to try and attack the coronavirus as soon as it's entered the body, okay? So if anyone knows the name of these cells, please do type it into the chat. These are the first white blood cells that try to tackle the virus on their own, okay? And if they can't tackle it alone, it then passes on to the second line of defense. These are a different kind of white blood cell that will take over the job of attacking the virus if the first line of defense was not able to get rid of it by itself. Okay, so basically the first line of defense, well done to those of you who guessed it, okay, it's called a phagocyte, okay. So a phagocyte is the type of white blood cell that attacks the coronavirus by wrapping its cell surface membrane around the virus like this, okay, it then destroys it like this. So this is probably what you have seen at your GCSE level version, okay. In the A-level version, we go into lots of detail on this process of phagocytosis and how exactly it happens. I'm soon going to just share another slide with you on this detail about this process of phagocytosis. But basically, the phagocytes are the first line of defense. They are the first white blood cells to attack the coronavirus as soon as we can see that the coronavirus has these foreign ID cards on its surface, these foreign antigens, which the white blood cells will not recognize as belonging to the body, and then the phagocytes will indeed attack them and gobble them up like this. So now we are just going to look a little bit more at phagocytosis. I'm just going to put this screen now on the screen. So basically, as I said, in GCSE, you probably will have covered phagocytosis to some level, where you talk about how a white blood cell called a phagocyte wraps its cell surface membrane around the pathogen. But at A level, this is the difference, the level of detail. Okay, you can see that there's so much more detail here. Okay, now the main thing, the main addition at A level in terms of how phagocytosis works is you'll probably notice this organelle called a lysosome, okay? A lysosome essentially is responsible for the actual destruction of the virus once the cell has taken it in, because it produces something called hydrolytic or digestive enzymes. These digestive enzymes break up the virus into millions of pieces. So basically, this is just to emphasize the difference between GCSE and A-level. Most exam boards at GCSE don't have this much detail for phagocytosis, but at A-level, this is the kind of detail that you can expect for one example process like this. Okay, so essentially, just to summarize, that's the first way in which the body gets rid of the virus. It's cells called phagocytes, which are types of white blood cells. They will gobble it up like this. Okay, so now I want to just have a quick quiz. Okay, once again, if you'd like just to type this into the chat. Okay, first question: how does the body recognize that there is a foreign pathogen like a virus inside it, which should not be there? And the second question: how does the virus attack your cell, and what does it do to it? Okay, so if I can just ask us just to write the answers to these questions. So you can either write this on a little piece of paper, or you can write it into the chat. Okay, and then at the end, we will go over the answers to all of this. Okay, so once again, the first question: how does the body recognize that there is a foreign pathogen like a virus inside it, which should not be there? And the second question: how does the virus attack your cell? What does it actually do to it in terms of what does it do to your cell? Yeah, how does it actually change your cell? So I'll just give us a few moments just to jot down the answers to those questions, okay? And then we're going to move on to the second part of today, which is talking about vaccinations and how that's related to all of this in a short while. Okay, so now I'm just going to now move on to here. So basically, we have just talked about what the first line of defense is, the phagocytosis. Okay, once again, like this. So as I said, if the phagocytes by themselves are not enough to fight the virus alone, then it goes to the second line of defense, which is a bit more tactical and a bit more specific in its approach. These are types of cells called lymphocytes, okay? Lymphocytes are another type of white blood cell. They make antibodies, okay? So I have just drawn here the antibodies underneath the lymphocyte. Basically, what happens is that the lymphocyte recognizes the antigens on the surface of the virus like this, okay? And according to the exact shape of the antigens, they will make antibodies which can match that particular shape of antigen, okay? So those of you who are on GCSE course, you probably have done enzymes as part of your course. So if anyone knows the word beginning with C, which we mean to say enzyme and substrate can fit together, it's exactly the same kind of principle here. The antibody can fit with the antigen because it's complementary to it, okay? So you'll probably recognize that word, complementary. The antibody is complementary in its shape to the actual shape of the antigen of the virus, okay? And so that is why, as we said earlier, each virus has a specific shape antigen on its surface. And it's this specific shape antigen which will generate specifically shaped antibodies against it. Okay, so each individual virus will elicit particular shaped antibodies to combat it. Okay, so antibodies are a little bit like your soldiers, a little bit like your army, okay, which will fight the virus for you. Okay, so just to summarize this slide once again, your first line of defense is your phagocytes will gobble up the virus like this, okay? When they recognize that they have the foreign antigens on their surface, they will know that they should not be there. These viruses do not belong to your body. And then your phagocytes will gobble them up and destroy them like this using their enzymes, okay? And once again, enzymes are molecules which will break up anything, okay? Or they can also build things up, as we also cover in the course. And your second line of defense, once again, are your lymphocytes. So your lymphocytes are a little bit more tactical than the phagocytes. Phagocytes literally just gobble up the pathogen like this, just gobble up the virus. But lymphocytes, they actually make specifically shaped antibodies against the virus like this. Okay, so now what I'd like to do is just to show you a little bit about how the antibodies actually work. Okay, I'm just going to add some things by drawing on the screen as well. So basically, as you can see here, what the antibodies will do, which are in blue, by the way, the blue represents the antibodies. The antibodies will bind to the surface of the virus, okay, and they will clump them together. This clumping together is something that we call agglutination, okay? Now, why is this beneficial for immunity? It's because if we just think one more time about the phagocytes having to eat up each individual virus, I'm just going to draw a few phagocytes. So imagine that we have one phagocyte there, which will go after this virus, and another phagocyte there, which will go after this virus. And then another one here, for example, which will go after that virus, yeah? As you can probably appreciate, the main advantage of clumping together these viruses is basically, it's so much easier for the phagocyte to gobble up the whole thing in one go. So rather than having 3 different phagocytes gobbling up, you know, one virus there, one virus there, one virus there, it's so much easier for one phagocyte to come and gobble up the whole clump, okay? So for that, it's much faster, and you only need one phagocyte to do that. Instead of having 3 different phagocytes, you only need one phagocyte to do this, okay? So that is one massive benefit of antibodies. They will clump the pathogens together like this to make them easier to digest by the phagocyte. Okay, that's one massive advantage. Another thing that antibodies do is basically they bind to viruses like this. I'm just going to draw that on here as well. They will bind to the viruses, and they will make the viruses more recognizable by the phagocytes. Okay, so essentially, if you think about it, it can be quite difficult for a phagocyte to recognize something as being self or non-self because the receptors are so small. However, if the antibodies bind to the virus in advance, they can make them more recognizable by the phagocyte. Okay, essentially, they can highlight themselves. They can highlight the virus for the phagocyte to notice it from a mile away, excuse the pun, and then just go to it and attack it. Okay, so that's how antibodies work and how they're so important. And once again, I thought that this will be a really useful slide as well, just to really explain the fact that each virus has its own unique shape, and its own unique shape has to be attacked by the particular shape of antibody, okay, just as we saw there. So imagine if we have this coronavirus once again like this. So basically, so as we just saw there, if the virus has antigens like this, then the antibodies have to be this shape, okay? Or if we have another kind of virus which has antigens which are not triangular, but rather they are circular, then our antibodies also have to be circular to match them. So that's why I thought that this would be quite a good comparison, comparing it to different padlocks having different keys to open them. In a similar way, different viruses have different antigens to generate different antibodies to break them apart, you can say. Okay, so now, and the final thing that I'd like just to say about this part is, so I thought that this will be quite a nice comparison as well. We can compare this to trying to open a locked padlock, okay? So once again, if you think about a locked padlock, phagocytes work by gobbling up the pathogen, like we saw, they just work by eating it up, okay, and destroying it. That's the equivalent of trying to break open a padlock using a hammer, okay? However, the second line of defense, the lymphocytes, they work by analyzing the shape of the antigens, making a particular shaped antibody in response. So lymphocytes work by actually going one step deeper, not just destroying the pathogen, not just destroying the virus like phagocytes, but they actually closely look at the virus and they look at the shape of the antigen. And then in response to that particular shape, they make the key, okay? They make their antibodies in response to the particular shape of the antigen, a bit like making a particular shaped key in response to a particular shaped padlock. Okay, so that's just one way really of thinking about the first line of defense, which is your phagocytes just destroying everything, and your second line of defense, which is your lymphocytes producing specifically shaped antibodies against the virus. Okay, now a quick quiz once more. Okay, and then we will come on to the last parts of this, which is about the vaccinations. Okay, because basically the vaccinations heavily relate to all that we've been talking about so far. That kind of gives you the background and the foundation to building a vaccination. So I would just like us to spend a few moments just to try to answer these questions. Once again, you can either answer them on a piece of paper or you can answer them on the chat, whichever one you prefer. So first question, what are the little key-like structures on the surface of the virus codes? A clue is it begins with an A. Why do antibodies have to be the exact same shape or complementary to the shape of those structures? And number three, how does the phagocyte do its job? Once again, I'm not looking for the A-level description at all for this. I'm just looking for a summary, what we talked about, how the phagocyte actually does its job. Yeah, three main things. So I'll just give you a couple of moments just to answer these questions. And then we're going to move on to the next part, which is the final part about vaccinations. Okay, so once again, you can either write these answers on a piece of paper or you can write them into the chat. But when we go through the Q&A at the end, then I can ask individuals to please share these. Okay, a few more moments and then we're going to move on to the next part. Okay, 5 more seconds, then we're going to move on. Okay, so part 2, we are now going to talk about why in general you can't get ill a second time from the same virus and how vaccinations work. So they are the same principle really. So basically primary and secondary immune response. So what happens is that, and once again, I'm just going to draw something on the screen. So the first time that you get sick from a virus, okay, so this is once again the coronavirus, it takes your body some time to react and to produce its antibodies against it. Okay, that takes time. However, the massive benefit of this is that you have something called immunological memory. Okay, this means that the second time your body sees the same shape, you can be ready for it. You already have recognized the padlock from the last time. You already have your memory cells making a whole load of antibodies so quickly the second time that you see it. Okay, you'll be ready for it the second time. Okay, that is why you can't really get coronavirus a second time. At the present moment, that is, okay, at the present moment, that is the conception, because there's a chance that it might change in terms of the structure of the shape, okay? But in general, that is why you can't get sick from exactly the same virus twice. It's because you already have built up your store of antibodies the first time that you saw it, and the first time that you see it, you're ill for about a week or 2 weeks while your body is making these antibodies. The second time that you see it, you recognize the virus, you recognize the shape of the padlock, okay? And that is why you can make your antibodies so much faster the second time that you see it, because you have immunological memory for the second time that you see it. So the second time that you see the virus, you can make a whole load of antibodies so quickly the second time, okay? And that is why, as you can see here, a large concentration of antibodies are produced so quickly. So basically, that is normally why a vaccination works, okay? What a vaccination does is basically you are injected with a fake version of the virus like this, okay? Now, the vaccination contains a virus which is dead or inactive, okay? This has the exact same shape and size of the real virus. Your body does not realize it's a fake virus. Your body does not realize that the vaccination is a fake virus. So your body will do what it normally does. It will make these antibodies against the vaccination. And the benefit is that you won't get sick in the meantime like you normally would for the real thing, okay? So the massive benefit is that the second time you see this, in other words, the first time that the real corona comes after you get the vaccination, okay? So essentially the vaccination has prepared you in order to make so many antibodies against this virus, so that if you ever see the virus, the real thing, if you ever see the real thing, your body will be so ready. Your body can then make a whole load of antibodies so quickly because you can recognize the virus. You will have seen it already from the vaccination. So you will be ready for the second time that you see it, which is actually the first time that you see the real thing. Your body will already have developed immunological memory against it. Your body will recognize it the second time that it sees it, and then your body can make a whole load of antibodies so quickly the second time that it sees it. Okay, that's normally how vaccinations work. Okay, now as you can appreciate, this is all dependent on the virus not changing its shape between the first time you get it and the second time you get it. In other words, whatever shape the vaccination has, this is kind of relying on the fact that the vaccination is the same shape as the coronavirus. Okay. But then what if you were given this vaccination and then a few months later, the coronavirus changes its shape, okay? So this is not a good idea because of course, then what will happen is that the first time that you get the vaccination, for example, so imagine that the coronavirus used to look like this, okay? And then of course, you will go through this whole antibody production, and then you will be ready for this exact same shape if it comes again, okay? But then if the coronavirus has changed its structure now, instead of having triangular antigens, it has circular antigens. Well, unfortunately, in this case, your immunological memory will not work anymore because basically you will not recognize this particular shape of the virus. This is why, unfortunately, you will no longer have immunological memory against it. You will not recognize this. You'll again have to be sick for a whole week until your body now caters for this shape, making circular antibodies. Okay, so just to summarize that, a vaccination is only effective if you have the exact same virus the second time round. If you have a different virus the second time round, you won't recognize it. If you do recognize it, if it hasn't changed the shape of its antigens, then thanks to your immunological memory, you can recognize it immediately the second time. You can produce antibodies immediately from the second time because you recognize it. But if the virus has changed its shape, something called a mutation, which some of you probably will have heard of, a mutation, then unfortunately, you will not have any immunological memory against it. So you'll not be able to recognize it. You'll again have to be ill for 1 or 2 weeks while your body now makes this new shape of antibody. Okay, so once again, so this brings us to the end of this session. So if I can just ask you one final question. If the virus has not changed, why can you not get ill a second time? Okay, so this is the final slide, this question, just to basically consolidate all of this. And then once you are ready, we can begin the Q&A. So thank you very much. Okay, so this now brings us to the end of this talk. And now I would like to introduce a student from the course, Simran Mohin. She is with us today, and she would like to say a few words about her experiences so far at the college in the course and how she's found everything so far, which will just help. And if you have any questions for Simran as well, please do ask. So Simran, please take it away. Thank you.
Thank you. Good morning, everyone. I hope you're all well and keeping safe. My name is Simran Mohin, and I'm a Year 12 student studying Biology A-Level along with Media Studies and Business Studies A-Level. I thought it would be a good opportunity to give you all my insight of Biology A-Level. So firstly, what do I like about doing Biology A-Level here at DLD College? I would say my favourite things about Biology A-Level are that I have an excellent teacher, which in fact is Rini. She engages with us and makes the class feel very confident. She does this by confirming we understand the concepts before moving on, which has really helped me to grasp the information clearly. As we are only 4 years into the new specification for the OCR Biology A-Level, even though we do a lot of exam practice in class for each topic, as well as a plethora of question practice from worksheets, the number of new specification papers are limited compared to the new of old. However, Rini provides us with extra exam questions from this resource called Zigzag, written by OCR examiners, which is a very beneficial resource as it is helpful for preparing for the new style papers, as we can see what questions are likely to come up and how best to answer them. My favourite things about the course is that the information we learn is so fascinating. The transmission from GCSE biology or equivalent to A-level is captivating. At A-level, you really start to dig deeper into the particular topic. Another thing I like about the course is the write-ups which we do for the biology practicals. In fact, I have a passion for writing, so putting my favourite two things together makes me so keen and ecstatic. Lastly, I also love our debates which we do in class. For example, on stem cell therapy. As you might know, it's a very controversial topic, so it's great to hear the varied opinions people may have, especially as it's an international college. Ethics is a big thing in our debates. Thank you all for listening to my insight on Biology A-Level. I will now pass it back to Rini for the Q&A. Thank you, guys.
Thank you very much, Simran. That's very nice to hear. Thank you.
Thank you.
Thank you, Simran, and thank you, Rini, for a really enlightening presentation as well. I haven't got any questions. I think you've been so thorough that everyone's just in awe of the level of biology knowledge and the session you've given. But I think maybe if you can go through the answers to the questions that you posed, because I think people have probably been writing them down at home, that would be great. And then I will invite, if anyone does have any questions, please post them in the chat now. And after Rini has gone through the answers to the questions, we'll be able to address those.
Great. Thank you very much, John. Thank you. Okay, so the answer to the first question in terms of what are the little key-like structures on the surface of the virus. So well done to those of you who said antigen. That's the name of these little structures once again on the surface of the virus. These little structures over here, yeah, they're called antigens. So these are the structures which our body recognizes as being foreign. Okay, so well done to those of you who correctly answers that as antigen. Okay, and second question, why do antibodies have to be the exact same shape or complementary to the shape of these so that they can bind to them, like we have talked about here? If the antibodies were not the same shape as the antigens on the virus, they could not bind to them like this. They could not group them together like this for phagocytes to gobble them up, yes? So that's why it's so important that the antibodies have to be the exact same shape, okay? For this principle, padlock and key have to work together. Similarly, virus antigens and the antibodies have to fit together in order for the antibody to attack the virus, okay? And finally, the last one, yeah, so, oops, no, sorry, wrong slide, sorry. Where was it? It was, sorry, there. Yes, so how does the phagocyte do its job? So well done to those of you who said that the phagocyte cell surface membrane will basically surround the virus and then it'll take it into itself, and then its hydrolytic enzymes or digestive enzymes within its lysosomes will basically digest it, okay? So, oops, and now I'm just going to check the chats. Okay, so I'm now just going to answer some of these questions on the chat. Okay. Oh, thank you very much, Tatiana, for the nice, kind words. Thank you very much. Okay, so now I'm just going to answer from Tyra Parker. So if the vaccine itself is not an exact replica of the virus, why is it effective? As if the actual virus mutates or changes, it's ineffective. That's exactly it, Tyra. Okay, let me now just quickly take us to this slide once again. So here, let me just make this full screen. So basically, when the scientists are making the virus, yes, they are using what they currently know about the shape of the coronavirus. At the moment, we can say it looks something like a particular way, like for example, like this, okay? So scientists are basing their vaccination on the size and shape that we currently know about the virus, okay? However, we don't know how quickly the virus will change itself, okay? We can only go by what is currently its shape, okay? That is why scientists, we are having to wait a few more months before we can be sure how often the virus changes its shape like this. We are having to wait months and months to see how often the virus changes its shape. So that, as you correctly said, Tyra, that if the virus changes, then what good is the old vaccination? That'll prepare your body for the old shape. It'll not help you for the new shape, right? So that is why, as scientists, we are having to wait months and months in order to register how quickly the virus mutates and changes itself, and therefore how quickly we have to keep making new vaccinations and again, new vaccinations to keep up with it. That's basically, by the way, why we can't have a vaccination for HIV, because HIV works like this. It changes itself so often, even when it's inside our body, it changes its shape. That's why we can't cure it and we can't have a vaccination for it. Okay, so I'm now just going to check another question on the chat. Okay, so I'm just going to look here. Oh, thank you so much, Elizabeth Bulanova. That's very kind of you. Your words are very, very kind. I really appreciate this. Thank you so much for your word. It's very kind. And so does anyone else have any questions about anything?
Rini, I just had a quick question from my side of things. Obviously, we heard from Simran talking about her experience on the course and the subject she's combining it with. What are the common subjects that students would combine with biology at, say, A level, and where would that lead in terms of their university destinations? What are you seeing on the course?
Okay, so I would say that taking chemistry and maths is significantly beneficial for biology, purely because there are certain modules in the A-level biology course, especially in AS, in the first year of the 2-year course, which are heavily related to chemistry. If one doesn't do chemistry, it's not a disadvantage, but one finds it easier to grasp the chemical structures of molecules quicker in terms of how they react a bit quicker than if they don't do chemistry. But it's literally just for 2 topics of the AS course, where chemistry is a benefit if you do that as well, because then you're more accustomed to seeing shapes of molecules and you're accustomed to drawing molecular reactions. So that's what I would say. If you take chemistry A-level in addition to biology, then it just helps you with certain topics, which if you do chemistry, you'll be able to grasp them like a little bit faster than if you don't do chemistry. So that's what I would say.
Brilliant, thank you. And what sort of university destinations are you finding students are heading to?
Oh, all sorts of places. So I think last year we had a couple of people going to Oxbridge. We also have, so a lot of our students go to universities in the Russell group, such as Nottingham and other places. A couple also go to Imperial College, where I went as well, and UCL, King's as well. So all sorts of different places. Some students go on to study in the United States. So all sorts of places, really. So.
Brilliant, thank you very much, Rini. And I believe yesterday, Dr. Daniel Woodley also gave a webinar on students' progression to Russell Group and London universities and the US. So if anyone wants to view that and see kind of where certainly DLD students, but also students from our Manchester and Cambridge colleges are heading, that should give you a good insight. I think as we haven't got any more questions, we'll kind of sort of round things up there and say thank you very much. We have been recording the session, so we should be able to send you a link to view the session, and we'll also send you Rini's slides as well, so you can have a look at those. One of my colleagues will be in touch shortly. But I think we'll call that it today and say thank you very much, Rini. Thank you, Simran. Thank you, everyone, for attending, and I hope you have a lovely day, everyone, wherever you are.
Yes, thank you so much, everyone. I really hope that you benefited and enjoyed this. And it's always my pleasure to answer any questions. Please feel free to always send me an email about anything that you'd like to ask. So thank you very much to John and to Simran for your contributions as well. And so I hope that was quite beneficial. And thank you very much, and see you soon.